Okay, now you can sleep better while listening to me. But it's me again, so I will avoid to make another presentation of myself. That it's even better. Okay, today I'm going to present a bit of theory about the ways that you can use to do the quality check on your high molecular weight DNA extraction. Okay, so basically there are three sets of tools that could be used. So one that you're maybe all of you know, it's the qubit. It's the qubit that gives you the concentration, so the final quantity of your DNA. And then you have, for the purity, you have the nanodrop. Somebody measure also concentration with nanodrop, but we saw that is not so precise as an instrument. But for the purity is the best one. And then for the fragment size, you have two options. Somebody told me yesterday that 20 years ago they were already using the pulse field electrophoresis, and I can tell you honestly that today is still the best technique you can use. But it is expensive in terms of time and of preparation. It took 2 days and then you have to run it, it's complicated. So sequencing centers started to use the fragment analyzer that can give you similar results, but is not as precise as the pulse field electrophoresis. So as I said before, I won't give you much detail, but you can ask, I will write in the report the details. The nanodrop give you two ratios. So is the measure of the absorbance of light of molecules. DNA is around 260. And then you have two ratios, usually 260, 280. It's about proteins and RNA. Because proteins and RNA have an absorbance around the 280 nanometers. So what you have to do is to have a ratio between the two of 1.82. The machine will give it to you automatically. If you are in this range, you have pure DNA, okay? And then the second one, it's about, let's say, organic substances. So it is still a DNA on the organic substances. And also there, you have a bit of a larger range in between 1.8 and 2.2. But this is just a bit of theory. Maybe if you need more detail, I can give it to you later. And then the qubit gives you the nanogram of DNA on a given volume. Usually it's nanogram per microliter. That is the most common used measure. And it's quite sensitive, yes. The only thing about qubit, it works with a fluorochrome. So this fluorochrome, when you put it inside, is stimulated, let's say, and if it emit fluorescence, it means that it binds the DNA. The problem is that it doesn't bind only DNA. The problem if you use, for example, magnetic beads and then you do an estimation of your DNA, it will be higher magically because magnetic beads do the same things, all right? They can bind the fluorochrome. And the best technique still, in my opinion, is the pulse field gel electrophoresis. It's a gel, as you can see in the picture, but it gives you, it alternates the directions of the pulse, the magnetic field. So it doesn't go only on one direction, but it goes in many directions, as it is shown in the picture. Why it does this? Because you don't have the smear. Sometimes you have a long smear and then a fragment of DNA and you don't know what happened. Sometimes it's only because the DNA within the gel is migrating too fast and you are basically breaking it, all right? But in this way, you can have the exact size of the DNA with good precision and without smears. As I said, my favorite, but too long. Let's go on the fragment analyzer. There is the latest technology, let's say, to quantify high molecular weight DNA size, concentration, and integrity. So there are 3 points that you can get back from it. Now here I brought an example of a genome that we are doing with ERGA Switzerland that the Rob group is working on. This is a mayfly in English, I suppose. Yes, it's a mayfly. It's a larvae of a mayfly. And this is what I did in the lab. So basically, when you run a fragment analyzer, we did this morning, we've analyzed some steps yesterday to don't keep you too much in the lab without a reason because it was like a one-person job. So what we did, we centrifuged the tubes that you gave us, all of them, even though you didn't have the DNA, the visible DNA. We centrifuged them for 15 minutes at 4 degrees and then we wash them with ethanol 70%. It was simply this procedure that we did. It will be in the protocol, but it's really simple to replicate, so we didn't want you to make it. And then this morning, Anastasia, not me, she did the qubit and the fragment analyzer. Okay, this is the output that you get back from sequencing centers. I know that sometimes output from sequencing centers are really difficult to read because you don't have time, right, to understand all of the points. Basically, on the y-axis, you have the intensity, the absorbance, and then you have the size on the x-axis. And then you have two peaks, one that is called LM and one that is called UN. It's lower limit and upper limit. Basically, these are known fragments that you feed into the gel to understand, to give a range to the fragment analyzer. All right? And in this case, when you work with high molecular weight DNA, we routinely use the upper limit at 200,000 base pair. So you are sure that you're going to capture it, okay? But the kit that you can buy, they say routinely, are the one from 1 to 60,000. So don't buy it. Don't go routinely. Okay, and then there are 4 characteristics that you can get from an electropherogram. So the number of the peaks, what would you expect in this case? Yeah, now you see it, but maybe it's not my best example. If you have more peaks, what does it mean? That you have different size of DNA. And if you are working with high molecular weight, you only want to see one peak, thin, single peak. And then the peak height, it gives you the concentration that you have in the sample. And yes, the peak size is give you the size. Okay, and then you have the baseline. What you want to see here that is not exactly perfect in this part here, yeah, it's a flat line. So if the baseline is flat, you have a good quality DNA. Okay, it's not fragmented, you don't have other groups of fragments, and it's perfect. And then the other output that you have is the gel, because when you run the fragment analyzer, you are also running a gel. And as you can see, in this case, it's not the exact size of the DNA. You have a range, you have a distribution, as you can guess. And the mean of the distributions were 46,000 base pair in this case. And usually for our, when we prepare, we use this to prepare hi-fi libraries, long reads, and it works perfectly with this size. That is not exactly because you see, you can have, under this curve, you can have different sizes of DNA. But the mean was this one, and when we have this means, for us, it's good. Okay, you can process and you can prepare the library, hi-fi library. Okay, and what you receive, it's also a file like this that is super simple to read. So you have the numbers of the peaks. In this case, we have the lower limit that is set at 1 base pair. This is the size. And the upper limit, as I said before, 200,000. And then this is the size of the mean of the peak, 46,086. And then you have the concentration, because the fragment analyzer tells you also the concentration that you can compare with the qubit, and it will never match, so good luck. I don't know why, but yeah, it's not up to me to solve this problem. And then here it gives you the size of the smaller fragment in base pair, and here the size of the larger fragment. It means that on the distribution, the extreme of the distributions in this case of Eperus assimilis were 6,105 base pair, and the max 130 bases, 1,000 bases. That is quite good if you need to prepare hi-fi. Okay, and then yes, this is the average, 50,000. That is good enough. And then you have the area of the peak, and with the area you can also estimate the concentration and the molarity. All right, but now let's go. As I said, Anastasia did the cube. Yes, Alice, we can do the question in the end. Okay, so I don't want to keep you here for too long. Thank you very much for yesterday. It was maybe a bit chaotic, but you all managed, but one group, you know why, all managed to obtain DNA. The bench number 2. So what I'm presenting here is the tissue weight. So when I dissected the thorax, we weighted it, all right? Because you always have to routinely keep all of the data if you want to improve your protocols in the lab. You cannot exclude any kind of data. So the tissue weight was 0.140. The qubit told us that there were 12 nanogram per microliters, and the good news is that we diluted in 300 microliters for a total amount of DNA of 3,600 nanograms. This is pretty good. And now, if I have to be honest, this is the lower limit that we accept to send the sample to the sequencing center or to us to prepare to do the library. So it was really good. Yeah. And then we have the bench number 3 with Elena and Alice and Roya and Sarah. I don't see where you are. Yeah, here. The initial tissue weight was 0.130, almost 5 nanogram per microliter. That is not bad, and 1,485. So no. This DNA could be used for everything now. I mean, except for yes. I didn't say it. And then for a total 1,400, that is good for other applications. And then the bench number 5, yes, there. So the tissue weight is 0.140, and I wanted to show you that it's not an issue of the initial weight, okay? No, I didn't say it. And then the qubit is 3 nanogram per microliter for a total of 876. That is still a lot. If you want to prepare a library, for example, for whole genome sequencing, it's more than enough. Later questions. All right, so these processes, for people that worked in the lab their life, sometimes I was going during my PhD to my supervisor and, oh, you did a library and it is like this and the other library is like that. Why? For wet lab people, it's really difficult to explain why, because it's not exact science what we are doing. So please stop to ask why and try together understand which are the steps that we can improve. But repeatability is never 100% in genomics. This is what I learned during my career. We would like to show that it is like this, but actually it's never like this. Rarely, I would say. Okay, let's take a look of the results of the gel. So unfortunately, we are not able today to see the electropherogram because the upper limit that we use was 60,000. It was available. I didn't brought with me the 200,000, but it's not an issue. You will not see your peaks, but as you can see, you see clear bands on the top. And this means that there are not much smears. So the bench 3, they have a bit more of smears. It means that the DNA is a bit more fragmented, but it's not it's really in a small scale. As you can see, if you go at the bottom of the smear, you have fragments of, let's say, 30,000. So it's already a good job. So yeah, you did a good job in the end. I was I was super happy this morning when I saw the results. And then I would like to conclude and then to accept questions with a huge thanks to the Aristoteles University researcher, especially to the one that I listed there, and again Anastasia that helped me this morning to produce the data. You raised the hand before? Okay, so this often happens because especially when you apply positive pressure, you will always have a larger surface on your curve. When you prepare the hi-fi library, independently for which one you prepare, they provide to you a kit to get rid of the smaller fragments. And this is the only way that you can compensate. It's not perfect, but you can still use because within there are larger fragments, and sometimes it's like a waste to discard the entire process only because the curve is larger. Oleg. Okay. 3. Well, no, come on. No, because it doesn't make sense because the fragment analyzer it stops at one point. All of the peaks that you will see will stop the median or the mean around 50,000. You cannot get more than this. It's in the protocols. They were flash frozen in liquid nitrogen. and then you take a petri dish, you put on dry ice, you have like, I would say 30 seconds to do the dissection. If you wait more, you can discard the individual. You cut the abdomen, you cut the head, you take it and you put it back on liquid nitrogen. Yeah. You take the part that you want. I suggest by experience, after 30 seconds is pointless because DNA the samples start to, DNA start to degrade too fast. It's in the protocol. If you have an insect, you can use the entire insect. I strongly suggest to avoid the abdomen because, we have bacteria and you will risk for small insects to amplify or to sequence, at least, many of the bacterial genomes. This is a problem that often occur. Yeah, and then you can get rid of them with bioinformatics, but if you don't have them, you minimize the risk of contamination. Oleg, please. Okay, electrophoresis will not work because agarose gel, it is too tight to allow to large DNA fragment to pass by. You're going to break it if you force it. So electrophoresis gel cannot be used to quantify high molecular weight DNA. If you don't have the possibility, I would go for concentration, as I said before. If you don't have a fragment analyzer, if you see that your concentration is higher than 3,500, you can give it a try with a hi-fi library preparation or send it to a sequencing center. Yeah, is the best way. And this is why this tool is great, because this costs like 30 euro to a sequencing center to perform the fragment analyzer. So it's not a huge expense and you can get a lot of informations. So if you don't have it in-house, I would suggest to send an aliquot. Okay, I didn't say cut, please. You never send high molecular weight DNA. This is a huge mistake that I heard in the Erga stream sometime. Oh, we are sending high molecular weight DNA. Because when it arrives, it's not anymore high molecular weight DNA. Okay? So, but if you have like a facility close by, you just bring an aliquot. And then, yes, and then with the, no, not frozen, room temperature. I left the samples when you finish the process, you leave them room temperature, and you can keep them in the fridge because the DNA is supposed to be, it's super stable in the buffer, and there are no enzymes cutting it because we got rid of them with all of the process. So in the fridge is fine, yeah. In my experience, then somebody would kill me, and as I said, is not each institution has his own habits and protocols. We have good results with this, we stick to it. When it will not work anymore, we will change it. Yes, it is possible, and this often happens. That's why we are going in a minefield. The quality of the reference genome should be adapted to the size and the quality of the DNA that you can obtain maximum from one organism. Okay, so when you contact or when you do the library yourself, you have a minimum requirements, and what they set is wrong. They set the ideal minimum requirement. We created libraries, hi-fi libraries with less than around 500, and it worked. But then you have to be really careful. You have to adjust, fine-tune the library preparation step of the hi-fi. I mean, you have to work on it. That's why it could be done, but it requires a lot of wet lab work. Yeah. It depends by the kit. Ultra low input refers to specific brand, but you can, yes, but you can. Yes, Spiros. No, I didn't say it. Yeah, but yes, sure, but when we grind, we put all together. So we can, but okay, so in this case here, I would, if it was me, I would work in a cold room first, not in the lab, like 4 degrees. So you work at 4 degrees and you work on dry ice the entire process of the dissection. You can take the tray, you can place it on dry ice like for 15 minutes before starting, and then you start to dissect on dry ice. Yeah, and this should minimize. It will not stop, but should minimize the degradation of the DNA. Okay, this is something. It's a super interesting question because we are used to take DNA out from the -20 and in, and each step is not good. Depends your application. If you have to prepare a whole genome sequencing library or a RAD sequencing library or a pool-seq, it's totally fine. You don't care. If you want to do a high-quality reference genome, my suggestion, personal, is when you're done, the day after, it goes to library preparation. And this will keep the flow, and you are quite sure that you are not introducing some dangerous steps, let's say. In the end, yes. I mean, for a longer-term storage, yeah. It is requested also in the manifest for the metadata, because if somebody would like to try, they have the possibility to retrieve the DNA or the tissue in an ideal world. No, no, never. What we with the protocol there is a specific equipment that you can use. So this is the procedure that you can apply to many tissue, and you see, for me, is the most important message. If you want, you can obtain it here in a train the trainer course. So if you want to replicate in your own lab, it's not impossible, and you don't need to have the latest technology to produce it. These are legends. If you follow the protocol, the chain of code, you can make it. Yeah.
So even with tiny, much smaller.
We did with the leg of an ant. But with a specific machine that helps you to don't lose anything. It doesn't introduce. You see that yesterday when you were working, you had some water that was freezing. So you had a lot of material and water was accumulating. With the other machine, this does not happen, and you are sure that you keep only the leg. And we were able to, it's not in such high quantities, but you are able to have a high molecular weight DNA from a leg of an ant. Yeah, I know that there are several species, but let's say an average sized ant. I don't say anything. I can tell you that it's really difficult to obtain RNA from the same individuals, to have enough material to produce high-phi libraries, high-C libraries. Yes, if you have an elephant, thank you. But, and then RNA. It's almost impossible, let's say, with one for each, I would say, but yeah, 3. But then, yeah, bioinformatics can kind of compensate a bit, but vertebrates, it's fine. But we don't only work with vertebrates. We work with plants, with algae, with insects, arthropods, really teeny tiny stuff. And it will never be possible, in my opinion. So you cannot expect, that's why what I said in the beginning, to have the same standards nowadays. Yes, maybe in the future it could be possible, but not yet. We are not there. Yeah. Thank you. Thank you.
You ready? Excellent. Okay, right. So yeah, as I mentioned, we're going to have a little bit of time to talk about indexing strategies for one-step or two-step PCRs. Everyone does it differently. There's no one-size-fits-all solution. So we're just going to go through the different options that are available and kind of some pros and cons, I guess, of the different options. As a background for PCR, and this will show you how little I understand about the entire process, I'm going to use the whiteboard here to just explain what happens in a PCR and then how we construct index primers and how that impacts whether you do the one-step or the two-step or everything else. So imagine you've got a little bit of DNA here, nice long fragment because we were so good at the genomic extraction there. We use primers, which are sequences that we generate artificially. We have a forward one and a reverse one. They match a specific place in the genome. So that's the forward one, that's the reverse one. And then we use a polymerase, which goes along here and fills in the gap basically between those two. We then have a cycle where we split the DNA and then we add primers again. And in essence, after a number of cycles, we have this nice one peak, no dimer, no other random fragments of other sizes that we've never heard of before. We have this one beautiful, well-amplified piece of DNA. In the case of what we're doing, this is obviously cytochrome oxidase 1 gene. And that's all great. It's obviously, we'll see in the gels, it doesn't always look like that. But traditionally, with Sanger sequencing, that's what you did. You then did another reaction, which turned this one fragment length into many thousands of fragment lengths of different sizes, each a base apart, basically. And you would send that to a sequencing center. They would run it on one of those ABI sequencing machines, and you'd get your beautiful traces back, right? Now, these days with next generation or third generation sequencing, we don't sequence each individual sample one at a time. We amplify them one at a time, then we stick them all together. We throw them onto a sequencing machine, and then we use bioinformatics to tease apart which sequences came from which well, right? And to do that, we have to, when we're doing the amplification, we have to add unique indexes to each sample so that we can bioinformatically tease them out. So where your primer traditionally was just a stretch of whatever, 15 to 25 base pairs, AC, GT, you know the usual ones. What we do with an indexing primer is we just add to the end of that primer another bit of sequence that we know. And so that can be between 7 base pairs or up to 13 base pairs. What we're using at the moment today is 13 base pair indexes that were designed by Amrita. So if I say anything incorrect, you jump up and fix that. And we use a combination that is unique for each well. So you remember your plate, it's, 8 by 12, so 8 down here, 12 that way. Each well, we put a forward primer that has an index sequence on it, and we put a reverse primer that has a different index sequence on it. And each well has a different combination of those things. And that's a one-step PCR. So there are advantages to a one-step PCR in that it's quick, one PCR, and you're done, ready to load into the library prep stage of ONT sequencing. The disadvantages are you have this index wobbling at the end of your primer, and that makes it less efficient, basically. Primers without indexes are more efficient at finding the template and amplifying. And there are ways around that. What you could do is you could do a PCR where you used a primer that didn't have the index, and then a second PCR where you take some of the product from the first PCR into the second PCR, and you use exactly the same primer, but this time it's one of the ones that has the index attached to it. And that just gives the second PCR a boost, because there are now multiple copies of what it should be attaching to, and they all have this end of the primer, so it should perfectly match. So that's one approach. It's essentially a two-step thing where the first PCR has no indexes, and the second PCR has indexes on it. You can also, the other disadvantage of this approach is that you have to order the primers with the indexes on them, right, if you're doing the one-step PCR. And so that's great if you're doing just insects and you're just using the Fulmer primers. But if you want to do crustacea and various other groups that don't work well with the Fulmer primers, then you need to buy a whole nother set of indexes with those primers. So there's a way around that, which is a two-step process. And what you would do then is you would do a PCR with a primer. So this is your forward primer, imagine, and then it just has a tail, which is another sequence that you know, right? And then after you've done your amplification, you'd have a product. So if you've got a reverse with the equivalent over here, you would have a product that has the tail, the primer sequence, the sequence of interest, the primer sequence, and the tail over there, right? So those are your little tails. And then you do a second step PCR, but you know what the sequence of that is. It's not unique per well, it's the same in every single well. And so you would have a second primer that matches that tail and then has your index on it over here. So that would be your forward that matches the tail. And the way, the benefit for that is these primers are cheap to order, and so you could have one for CO1, you could have one for 16S, you could have one for various different taxonomic groups, and then you would only need to buy one set of indexed primers, because then you've done your PCR1 and you've attached this tail, so your second PCR will use the same primer, but then have the different indexes. Hopefully that was not a very confusing way of explaining indexing, yeah? How much do they cost? I know that's a common question people ask. You buy them as regular primers. They're just a little bit longer than regular primers, so the cost is per base. About £10 is what we find typically for the 13 base pair long ones, indexes. And that makes about that much working solution of each primer. So that's about 150,000 PCRs for £10. So if you are making one plate's worth, you'll need 8, say, forward, and then 12 reverse indexes. So you'll need to buy 20. So it's about €200 worth of primers. But then you would be able to run 100,000 plates. Well, no, actually, you'd have to divide that a little bit. But you'd be able to run about 10,000 plates with that 200 pounds. So it really goes quite far. If you wanted to do 2 plates, so you could use the same 12 reverse. So we just do the same 12 reverse here, and then you buy another 8 forward indexes. And because the combination of that forward, say That one's 1, 2, 3, all the way up to 8. That one's 9, 10, all the way up to 16. You can still use 1 to 12 of this primer here, 1 to 12 of that primer there, because your combination here is 9 and 1, and your combination here is 1 and 1. So they're different enough that you can do that. That just means if you want to do 2 plates, you buy 12 plus 8, which is 28, yeah? So you 20 for 1 plate, 28 for 2 plates, 3 plates, you can buy another 8 forwards that are different, so then that makes it 36, and then you can do 3 plates quite easily. There is a different way of doing things. If you we'll be doing the ONT library prep as the next step, and in that, broadly, what you do is you take your amplicons, you add some proteins to the front of them, and you prepare them for sequencing on the MinION sequencer. ONT have various different kits that you can use. The kit we are going to use doesn't include any indexing steps itself, but you can buy kits where they have their own indexes, and in essence, you could have 1 plate of PCR indexes that you use for PCR, your one-step PCR, and then when you go to the ONT system, they have up to 96 indexes. So you could basically do 96 plates where you use this indexing and they're all identical, but when you do the ONT thing, you can separate them out with another layer of indexes. And then your DNA fragments would look like this. You'd basically have the ONT index 1, you'd have the ONT index on the other side here, and then you'd have your index forward 1, and you'd have your reverse 1 there, and then you'd have your gene of interest over there. And then bioinformatically, you would use software to just look at the patterns here and here and say, okay, if it matches index 1 and index 2, stick it into this folder on the computer, and then within that folder, if it matches file 1 and file, and forward 1 and reverse 1, then put it into a subfolder there, and that's your sample over there. That's a very terrible way of explaining bioinformatics. Yes, Glenn.
Quick question, I don't think I asked this before. Do you need any spaces on the couple of ends before you go to the index or something like that? No.
So we don't, it just goes primer and then index. Yeah.
Right.
Yes. Yep. So these were based on the R9 chemistry of ONT. So we bought these in 2019. We're still using them. And so at that point, the sequence error rate was too high for something like a 9-base index to be good enough. But now ONT is recommending, I think 10-base indexes are fine for ONT. The indexing with the ONT, yeah. It's better to use your own PCR kits, yeah. Yes. Of the ONT sequencing, yeah. Good question. So we do 3 plates on a flongle. So there's different types of ONT sequences. The smallest is the flongle. The one we'll be doing this afternoon is the MinION flow cell, which is bigger. We do 3 plates on a flongle, and that works fine. The folk at Eyeball in Canada, they're now in their R&D lab. They've got 48 plates on a flongle. So a flongle costs, including the library prep and all that sort of stuff, around about 100 euros. So you can imagine 100 euros divided by 48 plates equals 2 euros a plate. So 20 cents a specimen for sequencing, yeah? They think by the end of the year, they'll be able to do 96 plates on a flongle. And the way to do that, there's nothing really magical. All you do is instead of what we're going to do this afternoon, which is we've got our PCR, we're going to take 5 microliters out of each well, stick it into a single tube, and then carry on with the process. The way they do that is to try and balance between this well had millions of copies and this well only had a few copies. They balance that by quantifying the DNA, as Christian was talking about, seeing how much DNA is in each well, and then putting in the correct amount to try and balance the amount of sequence that you get out the other end. Yeah. But for our labs, we find 3 plates is just simpler. You know, not many people are working at the 48 plate a day or a week sort of level. Yeah. I mean, you could, but you're just adding more cost, right? I think we come from a background of Sanger sequencing where the most expensive part of the whole process was sequencing. Now that is on its head. The cheapest part of the whole process is sequencing. The most expensive part is plastic and your time. That is the big things. And so we got to change our frame shift. So I don't even think it's that important to do many more plates per flongle. I think 3 to 10 plates per flongle is way more than enough. At that point, you're costing 10 cents or less per sample to sequence. And the cost is the PCR and the plates, basically, and the extraction that we're doing. Even though it's cheap, it's still more expensive than the sequencing. So I wouldn't waste my time with qPCR. Bead cleaning can also be cheap, can be done quite cheaply, but again, like the cost benefit, you need to sit down and think, okay, well, are we doing 10,000 samples a week? Then we need to really shave the cents. But if we're doing 100 samples a week, 10 pence per sample or 15 doesn't really matter. Yeah. Any other questions while I check my notes quickly? The error rate for ONT sequencing, it's like Q20 at the moment, so that's 1 in 100? Yeah, 99% accuracy, 1 in 100. Yeah. But with the software Amrita's going to show you tomorrow, because the error profile for ONT is essentially random, that you only need a couple of copies to generate a very high quality consensus. Yeah, yeah. So like 5 to 10 reads generates a really good consensus. Yeah. All good.